my name is Taylor Brown, and this is my light and dark reactions diagram. Okay, we are going to start off, number one, in the light reactions. And this is my diagram. Alrighty, now let's start off with how these things kind of start. Alright, you have light that comes in, and it's going to strike a chlorophyll or a carotenoid. Now what's going to happen is that the electrons, after the chlorophyll absorbs that light molecule, it's going to excite these electrons and transfer them in their excited state all the way to the reaction center. And you see that electron that's right there? That's the reaction center. That's kind of where everything starts going. Now that electron is going to move from this photosystem 2, down the electron transport chain, through, it keeps going down and down and down to the photosystem 1. Let me skip back a little bit. Okay, and now that electron is going to go into the reaction center of this photosystem. And now, after it reaches this photosystem, it's going to keep traveling down the electron chain through ferredoxin and to this NAD plus reductase reaction. That is the enzyme that's going to allow NADP plus to accept that electron that moved down that chain and a proton and become NADPH. Now that is one of the most important products of this light reaction. Now, going back to kind of explain this a little bit more, these two bars you see, that is the thylakoid membrane. That's where a light reaction is happening inside the chloroplast. Okay? And this side is the stroma that is in the space of, kind of like the matrix of mitochondria, it's the stroma of a chloroplast. That's where the NADPH is being produced. Now this is the thylakoid space. That's the other side of the membrane inside a thylakoid. Okay. Now if we go back to photosystem 1, we talked about the chlorophyll electrons getting excited. And when that happens, it's going to split a water molecule into a oxygen. And it does this to have a higher concentration of hydrogen molecules on this side of the stroma. And we'll see that the reason for that is so these hydrogen molecules can cross over when the electron transport chain goes through these proteins. And they'll cross over to make this thylakoid space more concentrated with those hydrogen molecules. And we'll see why right here. Now this is the same thing that happens in cellular respiration. These hydrogen molecules get really, really concentrated up here. So the ATP synthase can use energy, and these molecules will pass through the thylakoid membrane, making ATP from ADP. Just exactly like it happened in cellular respiration. Alright, so that kind of explains the light reactions, and those are extremely important because they make NADPH and ATP. And why those are so important, we're going to see right now, because those are what makes the Calvin cycle move. The Calvin cycle is what's happening in the stroma, and what it's going to do is it's going to basically fix a carbon. I mean, it's going to fix a carbon, but it's going to fix carbon dioxide. And what we want this for is to make glucose, or fatty acids, or amino acids. Now how this process starts out is it's going to take a carbon dioxide that's let in through guard cells at the thylakoid membrane. I mean, not at the thylakoid membrane, at the chloroplast membrane. It's going to let in those carbon dioxide molecules from the outside, and it's going to use those to fix the carbon and make glucose. Now, how this happens is we take a carbon, we add it to something called ribulose 1,5-biphosphate. That's abbreviated RUBP. And this is added together through an enzyme called Rubisco. Now, what this does is this breaks down this ribulose biphosphate into 2 molecules of 3-phosphoglycerate. Ooh, that's a tongue twister. Okay. And with these molecules of 3-phosphoglycerate, it's going to use that ATP from the light reaction to make 1,3-biphosphoglycerate. Now, our whole goal is to make this glucose molecule, correct? So this is kind of just doing some conformation and changes in that molecule to eventually get this glyceraldehyde 3-phosphate, which is what we want. Okay, now after it reaches 1,3-biphosphoglycerate, it's going to use that NADPH from the light reaction, that one right there, and it's going to make glyceraldehyde 3-phosphate. Now this is exactly what we want from this cycle. We want to take one of those glycerol phosphates, take it out of the Calvin cycle, and use that for sugar, fatty acids, amino acids, what have you. That could either go to cellular respiration, it can make fatty acids, it can make amino acids to make proteins, plenty of things it could do. So then after we take one of those molecules, it's going to go back in this cycle to keep producing that molecule. Now let's talk a little bit more about how many. Now, in order for this cycle to work, you have to have 3 molecules of carbon dioxide. And that is because when we need a 5-carbon sugar to make this molecule, but we only end up with a 3-carbon to start with. So you think, how do we get that? What we do is we need this cycle to turn 3 times. So we take 3 molecules of CO2 with 3 molecules of RUBP, and that makes 6 molecules of 3-phosphoglycerate. And we come down 3 molecules of the 1,3-biphosphoglycerate. 6 molecules. 6 molecules of the glyceraldehyde 3-phosphate, and we take one of those molecules to make a sugar. That leaves us with 5 molecules, which is what we want. Or we want a 5-carbon, So 5 molecules of a 3-carbon structure. So you think, we want a 5-carbon sugar, but we only have a 3-carbon sugar. That's what makes this process so complicated. It's going to use many steps in here between, and it's going to change from a 5-molecule 3-carbon to a 3-molecule 5-carbon. It's very, very complicated. And that is called the ribulose 5-phosphate. And ATP, again, from that like reaction, comes in and makes this ribulose 1,5-biphosphate, the change in conformations that we want from this cycle. So that kind of sums up, let me show you a picture of my diagram. That kind of sums up the light reactions in the Calvin cycle inside and what happens in photosynthesis. Now, real quick, I want to just say that this doesn't happen without sunlight. Sunlight is the number 1 key here. Photosynthesis doesn't happen during the nighttime because of that light that it needs, and the Calvin cycle doesn't happen during nighttime either because the Calvin cycle needs these ATPs and these NADPHs from the light reactions, so it stops as soon as the light reactions stop making this product. So there you have it. That is photosynthesis, and I'm out.